I'm going to talk a little bit about development, bioconjugates, and I think maybe one of the themes that runs through this, and maybe it's kind of fitting for the day that we have, is that, part of my personal view is that it's a team sport, right? There's a lot of people that need to work together, come together to make things happen, and hopefully we'll paint a little bit of a picture about how we do that in our approach, and have a few maybe interesting kind of case studies of some of the challenges that we've come across along the way, and how we've overcome some of them. So hopefully there'll be a little bit of interest for everyone in the room. Maybe just to start off, if I may take a few minutes just to introduce, where Piramal sits in this space and what we can offer, what we bring to the table in terms of services around the bioconjugate space. So before getting into that, I have used the term bioconjugate specifically rather than antibody-drug conjugate, which is maybe the more recognizable term out there. And I guess the whole idea behind that is, a targeted delivery of a pharmaceutical drug to the point in the body where it's needed. It was a term that, maybe someone like, who was thinking well ahead of his time, Paul Ehrlich coined, that term, the magic bullet. It was a long time ago now when you think about it. It was 1913 when this concept came around, right? So probably, quite a visionary in the field of thinking about, what the possibilities of modern medicine were. And then if you think about, where we've gone, the steps we've taken since then, it took 70 years from that point, kind of coining that term until we got to the stage of running the first clinical trial for what we would recognize today as an ADC. So it was an anti-CEA antibody with a chemotherapeutic conjugated onto it. So that clinical trial didn't go very well. It didn't clear the hurdles it needed to. It took us until the year 2000 till we had Mylotarg approved as the first ADC. So first one on the market, it took a little bit of a hiatus and then came back again. It's still there as a therapeutic that's helping patients. So in that kind of period of time, I guess, it's going back like to the last century. But the whole field, I guess, of bioconjugates and ADCs has really taken off pretty impressively since that first approval in 2000. So, by my counting, today we're at 16 approved ADC drugs, all in the oncology space, which I think is really quite an interesting point. And we're seeing a little bit of an inflection away from that now. But it's a growing field. There's 16 approved drugs. As of 2024, so that's probably a little bit out of date now, there were over 217 different actual bioconjugate molecule modalities in clinical trials. And who knows how many more in preclinical development. So, it's a really growing, interesting field to be working in. We're really, and I'm certainly very, excited, very happy, very privileged to be working in the space. But I think, when I say, looking at all of those, medicines that have been approved, they're ADCs, but I use that term bioconjugate. And for a reason, because things are changing and they're changing quite rapidly. We were talking a little bit about this, over lunch. It's not just a case of taking an antibody and conjugating onto that a cytotoxic drug. It doesn't need to be an antibody, right? It can be an scFv, it can be a bispecific of some sorts. And what you conjugate onto that doesn't necessarily need any more to be a cytotoxic molecule. We're seeing things like oligonucleotides, peptides, conjugators that can, or chelators that can take on radioactive atoms, dual-payload ADCs. And I think it's been driven partly by, the success of the field itself, and we're trying to, find innovative ways to overcome things like, resistance to particular drug mechanisms or, improve patient tolerability. And these are all maybe some of the kind of creative ways that the folks that we work with are trying to, improve the outcomes for patients. And as part of that, I guess you're looking at different technologies that are used to make the ADCs and the bioconjugates as well. So we'll talk a little bit about them as well, how they fit into the process and maybe some of the challenges associated with dealing with them, and the fun that you can have doing that as well. So maybe just that's kind of placing, I suppose, where we stand, what we're looking at, in terms of where Piramal fits into this. So we offer, a full set of services from R&D, from development, all the way through to clinical and commercial manufacture of bioconjugates, and across that complex supply chain. So covering antibody production, payload linker production, bioconjugation, and sterile fill as well. So we have the capabilities and the facilities to support all of these development and manufacturing requirements. If I was to put a few numbers on that, maybe if you wanted one or two kind of key takeaways. So we've been in the business for 20 years. We're the first CDMO who started working supporting clients in the bioconjugation space. We're very proud that we were that first moving CDMO. Because of that, you'll find that we've got a lot of experience and a lot of expertise within our organization that you can utilize if you choose to work with us. In terms of some of those numbers, we do commercial manufacture for 2 of those ADCs that we saw on the list earlier on for the drug substance, and another one of them we do drug product commercial manufacture as well, and everything in between. So we supported 44 different IND filings for bioconjugates over the years, 2 successful BLAs that have been filed that we've supported as well, and we've run over 27 integrated programs across that supply chain network of our facilities. And I guess if I could call out maybe just your attention, some folks think of maybe an organization like Piramal as being maybe purely a commercial site or purely a clinical site. I think we've got that covered in the sense of, we're not very heavily weighted in terms of either clinical or commercial manufacture. We've been doing both for a very long time. So you'll find really a breadth of expertise across the development life cycle within our teams. And if I could take a minute or 2 extra of your time just to kind of call out maybe some of the highlights of the facilities that we run in the space as well. So when we talk about the monoclonal antibody facility that we operate, 4 GMP suites and up to 1,000-liter bioreactor scale is what we run there. So it's around about the sweet spot for what's needed for early clinical bioconjugate manufacture, and that serves us quite well as part of our Accelerate program, which is phase-appropriate manufacture of ADCs. So the question is, we do say up here on the slides that we do clinical and commercial manufacture. So for example, here for the bioconjugate drug substance and the drug product, we do clinical and commercial manufacture. And the question is, are the manufacturing spaces segregated, or do we do development and manufacture within the same space? So yes, we do have development and manufacturing facilities and rooms within the same site for both of these. So we've got our non-GMP development space. So non-GMP, any manufacturing, be it clinical or commercial, can be shared within the manufacturing suites that we use. So the way that it works out, so say for example in our bioconjugation facility where we've got 5 manufacturing suites, one of them is dedicated almost exclusively to commercial manufacture that's being done routinely. So it just kind of works out that way, but we can still use it for clinical manufacture as well if the space frees up within the suite. So the question is, if we have antibody production, for example, on the same facility as or on the same footprint as the drug substance production. So the answer to that one is no. So our facilities, they're separated. They're not all under one roof, which you may find from other folks. I'll tell you a little bit about how we operate that later on as we go through. But no, we don't do antibody production out of the same facility as we do drug substance production. They are separate facilities. So I guess the way that we've approached it is not so much to try and build under one roof everything, but more take in a sense, I suppose, best of breed, where the expertise and where the teams sit, that have the experience and the expertise. We built the facilities out around that. So question then on drug substance, if I mean conjugate ADC, yes. So I'm talking about bioconjugate drug substance. So I know some people may call the antibody the drug substance, for example. Yeah. So actually, as we talk through today, so when I say drug substance, I'm referring to conjugate drug substance. So so maybe let me just kind of finish giving you a brief run through the facilities. And so our Payload-linker facility can manage high potent manufacture. So it's something that's more of a specialized offering, but we have got suites there that can support high potent API, and typically do need prep HPLC and lyo capabilities for that small molecule HPAPI, and we have those as well. That particular facility, we've done some equipment expansion there recently because we've seen a lot of interest there. It's a US-based facility. We've seen a lot of interest running through that facility in the past 18 months. So we've had to bring in some larger equipment to deal with the interest that's been running through there. It is a full commercial facility as well. We've got over 30 FDA approvals out of that facility as well. So, you've got an experienced team that has seen, again, everything through development through to commercial manufacture. And then that brings us to our bioconjugate drug substance and our drug product facilities. So the bioconjugate drug substance facility, again, clinical and commercial manufacture out of there, 5 GMP manufacturing suites. We recently completed an expansion there, a $55 million expansion completed just over 2 years ago. And we've got probably industry-leading scale there, up to around about 10 kilogram batch sizes for bioconjugate drug substance, which is at the top end generally of what the industry is looking for at the moment, but we can support it all the way through. And then finally onto our sterile fill facility, just up the road here in Kentucky. It is it down? Okay, down the road. Okay, right. I'm going to stop trying to do US geography then. So just down the road in Kentucky then is where we've got our sterile fill facility. Typically liquid or lyophiles, for ADCs and bioconjugates. We can support both. Full isolator-based technology there as well for handling with the high potents. And that facility is currently undergoing a major expansion at the moment as well. So we broke ground there just about a year ago. I think we probably also broke ground once or twice after that for photo ops. But the building has been going on there now for over a year. One of our largest expansions, actually I think it is going to be the largest expansion in our network, and that is going to come online in 2027, making that a really large scale drug product facility for sterile fill. So, really nicely matched with I think what the industry is going to need by that stage. So there's our brief run through on Piramal. I'm happy to take any questions on that again at the end, but hopefully see a little bit of a picture as to how we fit in. So, right, Stu said, I've got a scientific background. I can't really stay away from that too long. I love talking about this stuff. So I'm going to tell you a little bit about, I guess, what we see kind of in this whole field in terms of technologies. We support some of that. So, if I was to start off, and just think about, well, what actually are we doing? When conjugate, you got to, find some bit of a reactive handle to link together small molecule and large molecule. And traditionally, I guess you can kind of bucket it into 3 kind of typical categories for how you make that linkage. The low-hanging fruit are to look at what's already there on your protein or on your antibody, so be those lysine residues, accessible cysteines, or more recently, technologies that have evolved using enzymes to modify specifically the antibodies so that you can put a reactive handle on them. So I'll talk maybe just a little bit through each of these in turn and think about some of the implications for them. So maybe starting off with the lysine conjugates. So, the first conjugation types that were used for making ADCs, relatively blunt instrument in a way, in that you've got lysines, randomly, scattered around the surface of the antibody. So when you do conjugation on a lysine residue, you end up generally with a fairly heterogeneous antibody-drug conjugate, which means that, you've got a little bit more work to do on your analytics. You probably got maybe slightly over usage of your payload. And you've got maybe a more heterogeneous product that takes a little bit more effort, probably analytically, than if you had a very homogeneous ADC. Now, however, there are some technologies that are out there now that do make use of those lysine conjugates, but they actually specifically target some of those lysines. And there's a nice example that we have later on about how you can do that with some peptide-type reagents. And I'll do a little bit of a dive into that later on. There's a little bit of a sneak preview there on the bottom. Maybe the next, I guess, kind of era, I suppose, of conjugation that came along then really is, using the cysteines on the antibodies for the conjugation. So be that, either a cysteine that's engineered into the protein, so requiring a little bit of engineering of your antibody, or utilizing the cysteines that are already there, those disulfide bridges, which there are 4 in a usual IgG1, and taking advantage of them. So usually the kind of chemistry you're talking about is a maleimide reacting with a cysteine on the surface of the protein. And I guess one of the things I think is quite interesting about this is there's often a lot of debate about once you form that cysteine-maleimide bond, it can reverse, right? So you can actually lose your payload off into serum or into the buffer or wherever. It's a reversible reaction. It's true. There are little process steps and tricks that you can use with a pH step that open that. Less likely to happen, but also, some of the actual approved ADCs are known to be, quite labile in this sense, but they're still very, very effective and they have a beneficial effect for patients. So, it's telling you a little bit that maybe ADC design is something still more of an art than a science, which I think is really, really interesting, the whole space. And I guess, one of the more kind of recent innovations, I guess, in this field, certainly in cysteine conjugates, is about the way that you can use specific metal ions. So, for example, you can use zinc ions, and when you add them to your conjugation reaction stages, can help you define a specific drug-to-antibody ratio that you get. So it's just little tweaks and little usages of chemistry that can help you get a little bit more definition in your product. So, I'll leave it on that, with the cysteine conjugates. But there's quite a lot going on. And then finally, talking about the enzymatic conjugations, the usual approaches, I guess, for these, on each antibody, you've got 2 glycans, and you can use glycan remodeling enzymes so that you can engineer a reactive handle onto the glycan. So similarly, you can use other enzymes such as transglutaminases or sortases. Benefits here being work with native antibodies, or in some cases, you need to engineer in a reactive tag. But I guess one of the things about these kind of technologies is, you got to be conscious maybe of the cost, not only of the reagents, because you've got quite a complex process. Your operations are being multiplied to be actually able to run the process. So they can be a little bit more challenging and a little bit more, or I suppose, less cost effective to run. But they bring with them their own benefits. So I guess doing a kind of a whistle-stop tour, through, some of the technologies that are out there. How do you deal with all that, right? Does it, our clients will come to us and, we're always amazed, at the kind of ingenuity and the different technologies that are brought to us to try and work on. And I guess that's one of the great things. And I guess how we deal with that is that you can't necessarily be an absolute expert and have done everything that you're going to get with. So we got to think about how we build resilience into our organization and into our teams so that we can deal with that. And it's more along the case of thinking about what are the transferable skills, what are the process steps that you work with. And this is kind of our approach to it, in that, yes, we build scientific excellence into our teams. You back that up with structure. You got to have the suite of instruments and the expertise to allow you to build the data and to interrogate the technologies that you're working with. But you've also got to have really excellent understanding of those common unit operations that you're using in the process. So any of those processes or technologies that I mentioned earlier on in the previous few slides, you will typically need ultrafiltration, diafiltration, large molecule chromatography. You need to be thinking about mixing, scaling up. It's the same unit operations that you're using throughout. It's just different configurations of them. So once you've mastered the unit operations and you know how to apply these, you should be in a good position to be able to scale up any process that comes your way. The other thing that we do at Piramal is that we try to preempt what the market technologies are that are coming forward. And we will do, through our R&D teams, proof of concept studies to make sure we're up to speed, we understand before they're actually brought to us by a client. So, a nice little example of that I'm going to talk through briefly now. So I've got one or two little kind of specific technical examples, and then I'd like to give you an idea of maybe what it's like to work with Piramal on an integrated program, how we approach it organizationally. So hopefully to give you a flavor of what it would be like to work with us. So in this particular example, I'm going to talk about one of those newer modalities where you've got an antibody conjugated with an oligonucleotide. So the process here, as we said, it's the unit operations and how you got generally to, do a buffer exchange, get your antibody into the right environment to be able to work, to be able to do the conjugation reaction on. and then a chromatographic purification followed by a diafiltration to get yourself into the right final formulation buffer. So nothing groundbreaking, nothing, really out of the ordinary there. It's just about understanding the product and how it operates in these unit operations. So for an AOC type product, in this case, we're talking about an siRNA, so about a 21, 22-mer double-stranded RNA conjugated onto an antibody. And the key unit operations that you need to think about how this operates through the process are on the diafiltration and on the chromatography steps. And we'll talk a little bit about them. So from a CMC, from a development perspective, with these kind of products, one of the things that can be a challenge can be bioassay. So you typically are going to need a potency assay for your product. And for a cytotoxic ADC, one of those historical ones, that's pretty easy. You just measure how quickly cells die at particular concentrations of your product. Determine cell potency or your product potency that way. If you're working with something that's an siRNA or an oligonucleotide, you need something a little bit more sophisticated when you're measuring maybe gene knockdown. And we're lucky that we've got a very long established bioassay team who can find the right cell lines with the reporter genes that are needed to be able to have a good supporting potency assay for a novel modality maybe like this one. I've also called out maybe here as well, just purely thinking about understanding the product you're working with. Typically, when you're doing your process development and even your release testing for an ADC, you'll use UV quite a lot, right? So for determining your protein concentration, for measuring your DAR, it's probably the simplest, most foolproof way of doing that. But you can run into problems if you do that with something like an oligonucleotide, because you see down there in the bottom left, you've got, bottom right, you've got overlapping absorption at the 280 nanometer wave. You now have got a very crowding, interfering absorption from your oligonucleotide. So you can get inaccurate readouts somewhat. So you got to think a little bit differently about how you approach this. So one of the ways that we would do that is we would use, say, for example, SDS-PAGE to measure your DAR1, DAR0, DAR2, whatever, rather than using UV, for example. And that helps you overcome those challenges through development. So I'll talk very briefly, just give you a bit of an idea of how some of this looks. Sorry, do we have a question? No. Okay. How this looks in an example. So here we've got antibody, small siRNA, conjugate the two together with a cysteine-maleimide type conjugation. And when our team got their hands on this from one of our clients, what we noticed was that actually we saw some unusual small little impurity peaks showing up in the chromatographic separation. And the question was, well, what is this? So, having that suite of analytical techniques really helps you to understand what's going on and then to help design your process and design your purifications appropriately. So what we saw here is that what we're actually looking for is this product or this molecule up here, where you've got one single siRNA conjugated onto an antibody. So what you've got is you've got the guide strand RNA, and then you've got the actual active single strand of the RNA molecule on the other side, which is then released, and that has the therapeutic effect when it gets to the cell of target. Typically what you find with oligonucleotide conjugates, interestingly enough, for whatever reason that is that a DAR1 is preferential to a DAR2. So my personal theory is that it's down to the charges that are attached on these, and that you get lower efficacy on the basis of having two oligonucleotides conjugated rather than one. So it's an interesting phenomenon, but it seems to be relatively general in the field. And what we found here is that this small little impurity peak that we're seeing chromatographically was actually the conjugated antibody, but where you're missing that actually active siRNA strand. So you got the guide strand, but it's only single-stranded. So this is effectively a non-active impurity that was in the product. And this was a carryover from an impurity in the oligonucleotide. So understanding that, though, it allows you to go on and develop your chromatographic separation so that you design this out and that you've got a robust chromatographic process that can, that can purify out any of these impurities should they appear. And I guess the nice thing about this as well is the way that we operate is we do run matched, in this case, single-use chromatography and single-use TFF systems in our development labs, as well as in our GMP labs. So you've got that full scalability through when you do develop the process. So we have, I think as Stu already mentioned, JF in the room as well, who have expertise from the payload linker and high potent perspective. So I'm going to speak very briefly about this, but I'm sure that they'll be very happy to take questions as well at the end. But from the point of view, I guess, of thinking about the various different payload linker technologies that get brought to us. So the idea is relatively simple. You've got your, your payload, which has got your therapeutic effect, and it's somehow got an anchor at one end and then a linker in the middle. And the function of the anchor is to stick it onto the antibody. The function of the linker is to release that payload at the right time. So you want it to be stable in circulation, stable in plasma, but labile once it gets internalized into the target cell. And I guess, to take a kind of a step back and to look at, where the trends, I guess, in the field are going and have gone, when you look back maybe from the 2000 through to maybe about 2015, 2020 kind of time frame, it really was, I guess, once Adcetris came out on the market using MMAE as its payload, this really drove, I guess, a lot of interest and a lot of, I suppose, imitation, I suppose, is the best form of flattery, in the industry for auristatin-based payloads. And what we've seen recently is that, there was, apart from literally a little purple patch, where pyrrolobenzodiazepines were in favor, where folks were looking for very potent payloads, and they still do have a use case in hematology indications, but things have shifted more towards low potent payloads, which are things like the camptothecins. And, the success of Enhertu has really driven, such a surge in this in the industry. So they all come with slightly different, I guess, chemistries, slightly different considerations, as to how you approach them. But there's definitely a frame shift, I guess, at the moment in terms of what is in vogue in terms of payload linker technology. And I'll just give you a few little ideas as to, how we approach some of these challenges and how we can utilize, I guess, some of the strengths within our organization to help you solve some of your supply chain and logistical issues here. So here's a nice little example of a camptothecin exatecan type payload, which was manufactured and developed by our team at Riverview here in the US. So the brief is to make the payload linker that you can see over on the right hand side. And the typical approach to this, and the approach that we take, is that your final high potent GMP steps, you need to have at least 3 significant chemical transformations prior to your final product to be able to get this through its regulatory filings. So you have to have those 3 GMP steps in there. However, prior to that, your key intermediates, they can be actually manufactured non-GMP. So it gives you a little bit more flexibility as to how you set up your synthetic strategy to make it, I guess, the most cost effective and make the most effective use of the facilities and the organization that we have here. So in this particular example, we had 5 chemical steps bringing us up to our key intermediates, and then the final 3 were performed in the high potent and GMP labs of our manufacturing facility. One of the things the team is always looking at as well is ways to engineer out the need for preparative HPLC purifications. This is something that can give you a bit of a cost headache and a bit of a challenge in your process. So the team were able to engineer in a few crystallization steps here as well along the way for those intermediates, which is really beneficial for the synthesis when you want to scale that up. And then maybe one final example just on some of the, I guess, the creative solutions, I guess, around payload linker manufacture that we have here. So this is an example that uses, I guess, the nice combination of, I suppose, the East and the West. So our Indian-based facilities for some of the non-GMP, non-potent manufacturing steps, but using our US-based GMP facility for doing the final steps in the synthetic sequence. So the team here actually designed the synthetic route in such a way that we had some key intermediates that could be utilized and then branched out for manufacture of different particular payload linkers that this client was looking at. So it allowed us the ability to stockpile and stock up large quantities of some of those key intermediates out of our Indian facilities, which gives you a time and a cost advantage, and then progress them through to GMP manufacture at our US facility as required for the process. So it's a nice way, I guess, in which we can utilize the organization and the facilities that we have that benefit our clients, in quite a few ways as well. So I'll rattle through these reasonably quickly, but there was one that I did want to call out, and this I think is really quite an interesting example, and an example, I guess, of just maybe how the field and the technologies are shifting. So this is an example of a conjugation technology which is technically lysine based, but is taking a little bit of a spin on it. And it presented us with a few challenges that we were happy to be able to see that we were able to resolve for our client. So this is where you've got a peptide-mediated conjugation. So I guess we're all probably fairly familiar, if we've done any antibody production, that there are affinity reagents that you can use to pull antibodies out of solution. So you can design peptides that are specifically going to bind to the Fc region of antibodies, and that's exactly what this technology uses. So you've got a modified Fc binding peptide. You just need a native antibody. The peptide will bind to the antibody, and proximity based, it will put a reactive group close to a specific lysine on the surface of the antibody, which will then get modified and give you a reactive handle against which you can conjugate. So it's a great way of being able to make homogeneous ADCs or dual-payload ADCs from native antibodies. And it's quite a nice technology, but it requires expertise across peptide manufacture, across bioconjugation, across high-potent small molecule manufacture. So it's making that supply chain even more complex than it normally is. But we were nicely positioned to be able to help the client out on this one. And I'll talk you through maybe just briefly some of the challenges that were presented on this. So that peptide was a 34-mer peptide. It turned out to be aggregation prone on solid phase peptide synthesis. And to make matters even more interesting, it had a disulfide that had to be installed post-cleavage from the resin. On top of that, there's a labile ester modification on the peptide as well. So it's not just a straight up peptide. So there's quite a bit of complexity in this one. Separately, the payload linker that was being used in this case had a 4 amino acid linker on it. And there were stereochemical centers in both the linker and in the payload that you had to be careful about maintaining that stereochemical integrity during the manufacture as well. So, like I said, a complex supply chain, some complex chemistry, some complex reagents. And I guess we were able to utilize a facility that we also have which specializes in peptide manufacture to be able to support this product in full. So across the peptide, the payload linker, and the bioconjugation, everything came together quite nicely. So the team sport coming into it again. And the approach that the team took here was more of a hybrid peptide synthesis approach. So solid phase peptide synthesis, but we looked at particular resins, low loading on the resin so that you don't have any aggregation occurring as you build up the peptide chain on that resin. And then optimizing the solvents, the wash steps, and the coupling stages along there as well to make sure that you get a good process, a good yield, and you have a scalable process. Because the important thing about this is that you can do it at a relatively small scale, yes, but you want to use this for GMP manufacture, you're going to need to be using quite large quantities of the peptide, to be able to enable that conjugation. So that was what the team did, how they engineered the process for manufacturing the peptide. And then finally, just a global deprotection where we were able to take the peptide off of the resin and free up those reactive handles at the same time. And this was all enabled then or supported by analytics along the way as well to be able to look at it. So, a nice usage, again, the facilities that we have at our disposal, but for a very specific use case. And I'd be really interested actually to see how these technologies take off. I think that this is one actually that might have some legs to it, and we'll see a little bit more in coming years. So I'm conscious I'm probably running a bit over time, I'm not sure, but I want to give you maybe just a very brief run through and an idea of what it's like to work with Piramal on, you know, a complex project like this. So I'm sure anyone sitting in the room is going to be acutely aware that, you know, ADC manufacture is a fairly complex exercise. You've got a lot of different specialities, a lot of different facilities you need to manage. It can be like herding cats, I'm sure, if you're working with multiple different organizations for all of these different parts of the supply chain. So we try to take some of the pain out of that with our Accelerate offering, which is specifically focused on simple, easy first manufacture of your first GMP batches to support clinical trials. And that's across the entire supply chain. So we support that across antibody production, payload-linker production, bioconjugate drug substance, bioconjugate drug product. And we say that we will do that guaranteed delivery, fixed price in a 12 to 14 month time frame. Right? So that's our offering on it. You know, hopefully that's something that would be useful for folks. But I'm going to tell you maybe a little bit more about how that actually operates, which I think is maybe where the real value comes in, because it's not just having the facilities, it's how you operate them. I think you'll see now, you know, bioconjugates are, you know, they're hot, they're interesting, everybody wants a bit of the action, certainly from the CDMO space. Everybody is saying that they can do it now, and they're all saying that they can do it integrated. It's not just purely about having everything under one roof. You know, I think we had that comment right at the outset. It's about how you utilize the facilities and the teams to make things happen in concert and have it all come together effectively. And like I say, you know, Piramal has been doing this for 20 odd years, so we know quite full well how to do this. But it's not just about the facilities, it's about having the expertise, utilizing that, communicating effectively both within and between your teams and with your client, and about how you set up your organization to be able to deal with this kind of complex supply chain. So I'll give you a little bit of a window into our approach on that. So these kind of programs to work effectively, you need at the least 3 elements of control across your organization. If you're not thinking about how you manage and integrate quality, project management, and technical expertise across your program, you're probably going to run into problems at some point along the way. And we've set up our organization so that we have, for our bioconjugate and biologics network, we have a global quality head who assures alignment of our quality system across all of our facilities that operate in our bioconjugate network. If you are working on an integrated program across multiple of our facilities, we will be appointing an integrated program manager to your project as a single point of contact from a program management perspective, and they will interact and coordinate the activities of our project managers at the specific facilities. And then you'll have someone like myself, or JF, or Brad, or John from our science collective who will align technically right from the outset of the program until the execution at the end. So there's our 3 kind of levels of control that we put over the program for you. The eagle-eyed amongst you will probably have noticed that we operate across multiple geographies, right? We're under one roof, okay, but we do have all of the capabilities and the facilities that you need to effectively run this program. So for us, part of doing business on a daily basis is how we manage the movement of materials between our facilities. We have to be able to do that reliably, effectively, safely to be able to operate. And it's something that we've put a lot of effort into as well. So we work with AIT Pharma Freight. We've had a long-standing relationship with them over 8 years that we've been using them to commercial and clinical drug substance between our facilities, between continents. And we've got a strong working relationship with them. They are certified and audited partners of ours that we use in these transfers. So the way that we set this up is that any movement of materials, be that samples, be that clinical, be that commercial material between our facilities, that's something that Piramal organizes. We've got dedicated logistics teams and supply chain teams who will manage that, organize that, and who are familiar with the importation, exportation regulations in all of the geographies in which we operate, in which we move materials between. So we've got a very experienced team who know what they're doing. They've got all the procedures in place, and they manage a lot of shipments between our facilities. So to give you an idea, we're often asked this question, well, how many shipments have you made? How often do these things go wrong? So within our bioconjugate network, in the past 18 months, we've had over 20 shipments of GMP drug substance between our facilities for programs that we've run for clients. All of the batches that have been shipped, the delivery time frame has been between 3 and 7 days, with averaging of 4.2 days for delivery. None of those GMP batches have been delayed. None of them have been held up in customs. Everything has gone smoothly for all of the GMP batches that we have shipped between our facilities. So it's a question we're often asked. Yes, it's a risk. We know it's a risk, but we need to make sure that that happens smoothly, and that's why we've put in place the organization to make it happen. What I should say as well is because we operate under a single quality system across our sites, we can also do shipping under conditional release, which gives you a time benefit as well, as opposed to working with different organizations and different CDMOs. So that's an added benefit, I guess, that you have for working with us in those situations. So to give you just a little bit of a, final bit of a brief, I suppose, on how, we operate these things. I did say, you get your technicals involved right from the outset. And I did say as well, we say guaranteed delivery and a fixed price. So, one of the things that, you would be surprised how easy it is to happen if you're working across multiple CDMOs is that at some point someone will say, do you know what, we need a bit more antibody because we don't have enough drug substance to support your drug product development. So actually we're going to have to make another batch. Sorry about that, but it's going to take a bit of time and it's going to cost a bit as well. Right from the outset, we will work across, so folks like JF, myself, John, we work with our technical teams. We plan out exactly what's going to be needed, when, what grade of material is going to be required, how much is going to be needed for your stability studies, how much is going to be needed for your release testing, how much you're going to need yourself. It's a very clear understanding right from the outset of how much material is going to be needed, where it's going to go, what it's going to be used for. So there should be no surprises for you as the program unfolds, where we say, actually, hang on, sorry, we need another batch, sorry. So, we try to put that control in right from the outset, from when you get the proposal with us. And, a very brief, example, I guess, of how that operates. Across a recent program that we run, where we did the antibody production, drug substance bioconjugate production, and the sterile fill finish. This particular program is a phase program. It was interesting in the sense that it was a non-cytotoxic payload. It was a non-oncology indication, which meant that we were looking at something slightly different. Yeah, so slightly higher production volumes, and it was a higher concentration liquid fill, right? So 150 mg per ml formulation, which is quite high for an ADC, considering they're typically around maybe 10 to 15. So came with a few challenges. Some of the innovations that the team brought in to help this one run smoothly. So our antibody team, they did a great job actually on optimizing the feeding strategy for the production of this antibody. We got up to 9 grams per liter, so supply was never an issue in this one. Actually, what we found is that we were delivering those batches of antibody typically 2 to 3 weeks ahead of when they were scheduled. So we weren't right on the ragged edge in terms of timelines on this one. They gave us quite a bit of buffer right from the outset, which was great. From a drug substance perspective, because it's a high concentration, we had to develop the formulation. We added a few novel excipients, which helped us to get that concentration up, keep the product soluble at 150 mg per ml. And because you're doing your final formulation at 150 mg per ml, even though you're doing maybe 2 kilogram batch sizes, you can have a significant loss in yield in the hold-up volumes at the end of the process because you're at such high concentration. So we implemented single-pass TFF to minimize hold-up volumes and make sure that we got good product yield out at the end. So just a little tweak, but it makes quite a big difference to the output at the end. And then finally, from a drug product perspective, because of the high concentration, viscosity was a little bit of a challenge on this one. So we had to optimize the needle diameters and the pump speed to make sure that we were getting reproducible fill on the drug product. So just a few of the little tweaks. It seems relatively straightforward when you read it off the top, but just little tweaks that help things flow smoothly, through the process. So just to wrap up and to say, thank you very much for joining us. Thank you for your attention. If there were a few takeaways that maybe you take from this, hopefully you will have heard that, Piramal's got the history, the experience, the capacity and the facilities to be able to support you all the way through your clinical and commercial manufacture for bioconjugates. Hopefully you'll see how we organize ourselves in terms of building the resilience in our technical capabilities. And I think because of the way the industry is going, you need to be flexible. Flexible in terms of how you approach technology, flexible in terms of how you approach the organization and the delivery of your projects. And that's something that we try to take on board and take with a pride in being flexible for our clients as well. And then finally, in terms of the integration and the organization, we do spend a lot of time focusing on how we communicate internally and how we communicate with our clients and how we organize ourselves to make that routine part of daily business. So hopefully, it's been a useful session for you guys. Really appreciate you taking the time. Thank you very much, and take any questions if there are.
So back to the page we're going over the shipping.
Yeah.
It's the most critical part in the process, really, is your intrasite.
Yeah.
So is this, you do intrasite shipping, or do you ship on behalf of the customer and then absorb the cost of insurance?
So any of the shipments between our sites, we will organize, we will manage. We do have our own insurance, right? These things are typically, part of MSA as well, so there will, I'm sure, be some degree of negotiation, which is going to be above my pay grade on some of that. But in terms of organizing the shipments, yes, that's all on our team. If you want to ship material outside of our organization, our team can organize that for you as well, but we need to take your instructions, of course, on where it needs to go in your instructions.
Okay, thank you.
That's a really interesting question. So there, if you look up the scientific literature, you'll find, it's out there. Public space. You'll find a few papers on it. I'm happy to share a few with you if you wish. But at the moment, my understanding is that there are two different accessible lysine residues that have had these reagents engineered for them. So that you could put on, say, one reactive tag on, a pair of lysines and then a different reactive tag on another pair of lysines. So you could do, for example, a DAR4, right? Or you could do a DAR2 and a DAR2 with another payload. Or there's nothing to stop you from designing, say, a branched linker that you put on so that you can DAR8 or a DAR12 ADC. So that there's a lot of flexibility in there in terms of how you design the linkers as well as which conjugation sites you pick. But at the moment, I suppose the simple answer to your question, there are two different lysines for which you have these reagents that can react on. We have a question up here.
Just the antibody production, who may just need an antibody or a fusion protein or something like that, which may or may not be conjugated eventually. Where does that happen, and what are the capabilities for that?
No, but actually you've raised a really good point, right? Because what I presented here, is, I guess, a full spectrum offering. Now, there are folks who don't need that or don't want that, right? Or, they may already have in place, say, a supply, and like that, they just want to work with us maybe for an antibody supply. So treat it as something of an a la carte menu, right? Our antibody capabilities are big. So that's where we have facility set up. In terms of scale, I think you'd asked about the scaling on it? No? We do 1,000-liter bioreactor scale.
Would still have in India, or would transport it to the US or?
There's a small filling capacity in our antibody facility. Is that right, Dane? It's more. So we do some CGT vaccine work out of there as well. Scale fill. There's still food there, guys. So okay. Well, maybe we'll wrap up. Thank you very much.
Yeah, somebody asked about slides. We'll distribute the slides afterwards. And we also recorded this, so it'll be available as a webinar at some point.
Yep. Thank you.